Hello, everyone. Eric. How's it going? Today we're going to be talking about one of the first questions on the study guide and a couple of other things too. But really, it's the different structures that protect microbes from desiccation. That's a word, Professor. Antibiotics and predators. The three big things are going to be the glycocalyx, the flagella, and the fimbriae. These are found on prokaryotes and some eukaryotes as well. We're going to have cell walls and cell membranes as well. But some of the first things that we're going to talk about are the glycocalyx, the flagella, the pili, and the fimbriae. Man, that light's a little bit too not there anymore. How about that? We do less light. Okay. And so there's two different types of glycocalyxes, but actually, yeah, there's two types. First type is a rigid protein capsule. The other one's a slime layer. And the rigid protein capsule is made of proteins, glycoprotein. When you say glycocalyx, think of sugar, glucose, Krebs cycle, or Calvin cycle if you're a plant watching this video. How's it going, tree beard? I don't know, it's on the slide deck. Streptococcus pneumoniae. We underline species of microbes that are written. And then we've got the slime layer, and these are lipid based. So we've got glycolipids, and there are still some glycoproteins in here. There's also some polysaccharides. Something that's interesting, right? These are slimy, so they're sticky, but they also kind of shed, so like, how does this affect how it can be protective against stuff? If you are like attacking something and it's like Nickelodeon slime and just kind of like slimes right off and you slip and slide and you can't attack it, then that's how the slime layer protects it. Then you have the rigid one that's like wall and not doing anything. So, granted, if you exert enough force, you can break down. Because these things aren't, like, they're kind of like wiry, kind of like tree branches. Molecularly speaking, kind of reaching out and pushing things away or pulling things forward. Depends, right? But this is speaking protect. Flagella, they're little propellers. You've got three different kinds of compositions of them. You've got one that's like, okay, here's your cell. That's a shitty color. And you got this one, you got one of them. And then that's one kind. The second kind is, you got another one over here. I lied, there's four kinds. You can have it like that, or you can have it third iteration. Fourth iteration. And these are all propellers. And these do vary in eukaryotes and prokaryotes. In eukaryotes, they're going to be more advanced and they're going to have many structures inside them, actually even organelles. They're going to be made of centrioles, and those are those 9 sphere things. While these are going to be individual protein structures. So that's something to keep in mind. Next, we have the pili and the fimbriae. And the pili, kind of dish. Short, prickly around the cell, they kind of are everywhere, and they help stick to stuff, like a biofilm. We'll get to that in another lecture. Or just anything, kind of like false feet. And then you've got fimbriae, which are longer versions of those, but they're not necessarily flagella. They don't, but they can have pili on them, and they also do like to. All right, but they can share DNA. One of these little fimbriae touches you, and it's like, oh wow, I want to give you some of my prokaryotic DNA. It very well might. I get some prokaryotic DNA. Become unicellular. And now you've got in brief, 3 structures that protect all the cells: glycocalyx, flagella, and the pili. Because also, if you're being attacked, what's the best way to you're gonna be gone now because you're gonna be me. You just put your DNA in them, and now they're making your DNA, and then they're you, because clones, colonies, maybe. I don't know if it's possible or not. That'd be a cool way. Offense is defense, maybe. And now we're going to the wall, cell wall, prokaryote or plant. We don't have them. We have membranes. We have membranes over there. Next slide. But something that we have on the cell walls is we have techoic acid. And these are for gram positives, right? And we see those in this little space right here as the techoic acids. And sometimes we'll have acid fast gram, which is basically we'll have an additional layer of mycolic acid right here that I didn't draw because I forgot about it and I'm running out of time because I have to go somewhere. Very simple. Gram stain, gram positive, because it's just like a wall. I don't have any saffron. No way. It'll be purple, not saffron. Well, this is gram negative, right? And so this is the more complex thing that's going on here, in my opinion, because we've got a liposaccharide, which is embedded to form the outer membrane. So it's on the outside of the wall, right? So it's kind of, I'm gonna say something fucked up. And it's mounted to peptoglycans, and it anchors with murin lipoproteins. And so what that happens, right, is here you have this plasma membrane, right? So rewind it a little bit. T acids, right? That's what these are. They're anchoring this wall to the membrane, to the plasma membrane. Over here, we have something, and those are murin lipoproteins, and those anchor through the wall. I draw this right. Yeah, kind of. So this is the cell wall, right? So, and it very well might be as thick as this, right? This wall, but right. So now you've got another plasma membrane. So you've got another one, an outer membrane, right? And this outer membrane uses these murin lipoproteins to mount onto this plasma membrane through the wall. And then this membrane has things called liposaccharides that are mounted to this outer membrane. And this outer membrane, these liposaccharides are constituted of 2 parts: the lipid A and the O antigen. A antigen? Oh. This is a pour-in, right, to let stuff in and out. Of the outer membrane, right? Because now you've got another membrane, it's got to be able to do all the stuff. Can you see that? No, you can't. It's going to fall over, but I'm going to do it anyways. This right here, these are just membrane proteins. And like if this is in a prokaryote, this is where the electron transport chain would be, which is kind of interesting to think about, because then you would have potentially you'd have hydrogens and protons being pumped into this periplasmic space. So maybe the wall is holding it in place. But if you're in again, this is for that's a gram negative, so I don't know about gram positive. Next. What makes up a cell wall, right? Because we talked about Both of them have the wall, right? That's an essential thing. If you're going to have a wall, it's going to be a wall, but there's going to be two different kinds. It's going to be a positive, gram-positive one, and gram-negative. And kind of chicken or the egg, from my understanding right now. You've got similar wall structure, right? But is that because the membrane has changed the structure of the wall on a gram-negative cell? Or is that because the gram-negative cell has had a different wall structure to incorporate the membrane? Theory. But the wall is the wall is the wall. It's got two important basic components, dimers, you might say. And these are N-acetomuric and N-acetylglucosamine. Amine, protein, glucose, sugar, uramic acid, NAMs and NACs. It gets a little confusing from here now because that's about everything that's in common is these two dimers. And that's it. And then so in gram positives over here, you're going to have pentaglycine protein chains, so 5 protein structures linking these 4. Because this is basically a huge, it spreads out in many different directions as you saw here, right? You got this, you orient, rotate, it's just that one little thing, right? Maybe, if that. There's many compounded in there. Gram positive still. So you've got these 4 protein chains that connect down onto this main set, right? And mind you, you're gonna get, it's gonna go like this, right? Don't worry. Where's my red? And that's why professors don't do this. Actually, you fuck around. Like, just don't draw good. Right? And so it's expanding out many different directions. And so you got this 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 5 protein bridges that are connecting these rows together, making this sheet, this wall. And then you would have, if you were gram positive, you'd have a tetroic acid. Mind you, this is 3-dimensional shape going, anchoring it down. 5 protein structure. 4 protein structure, right? So it's connecting down from the nag down to the 5 to the rest, right? So only the nags cross-link. The NAMs are alternating in between the nags. Dimer. Gram negative. You've got nags and NAMs. You've got those 4 protein structures that are holding the nags down onto the bridge. But now it's not a 5 protein bridge. It's a direct cross bridge. So it's close. And again, we would this time have, I don't know. Actually, I don't know. The diagram doesn't show it, Professor. You would have urine lipoproteins at the very least coming down on it like that, anchoring down this outer membrane over here, right? This is the outer membrane. This is the inner membrane. Thank you.